So let's talk about inheritance. Now, there's quite a bit here, so it will definitely go over more than one video. So what I'll do is I'll do a sort of a part 1, part 2, maybe a part 3, and I'll try and give you an idea of the stuff that's going to be covered in each of the videos. Here's the syllabus objectives. Whilst there's really only 2 syllabus objectives, and it says take 3 hours to do this in class, they are sort of jam-packed. So again, like we've done in the past, make sure you go back and have a look at the syllabus objectives at the end, and also the guidance material to know what you do need to know and what you don't need to know. All right, let's get into it. So, of course, inheritance, what we're talking about here is, organisms inherit the genetic material from their parents. So we get half the genetic material from mum and half the genetic material from dad, and that's in the form of chromosomes. So we end up with 2 homologous chromosomes for each of our chromosome numbers. And so each of these homologous chromosomes have the same gene, but they may have different versions of the gene. And that's what we call alleles. So alleles are the different versions of a gene. So the process of inheritance is all about which genes get expressed and which ones don't. So that's what we're talking about in this video. You probably have remembered dominant and recessive alleles from the work you did in year 10. Dominant alleles, they're the ones that we express with a capital letter, and they are always expressed when they're present in either the heterozygous or the homozygous states. So let's just remind you what they are. Hetero means different. So, and zygous, we're relating to the zygote. And so heterozygous means different alleles on the homologous chromosomes. Homozygous, homo means the same. So homozygous means the same alleles on the homologous chromosomes. So with the dominant alleles, that's the one that's expressed whether it's in the heterozygous or the homozygous states. And the recessive allele is only expressed in the homozygous state, where both of the alleles are recessive. We express the recessive alleles with a lowercase letter. So the genotype is written as 2 letters. So this is homozygous dominant, capital A, for a particular gene or trait that we have assigned the letter A. Now, so we use the same letter for both the dominant and the recessive. We use the capital for the dominant. We use the lowercase for the recessive. So homozygous dominant is going to be capital A, homozygous recessive is small a, or lowercase a, and the heterozygous state is capital A, lowercase a, and we always have the capital letter first. And of course, in the heterozygous state, it's going to be that dominant phenotype that's expressed. Don't forget we use the same letter for the dominant and recessive. So the syllabus objectives is all about predicting what the genotype ratio and the phenotype ratio will be of a cross of parents. One of the ways we can do that is using a Punnett square. It's a really nice summary diagram and a nice way for us to actually do our calculation. Okay, so when you use Punnett squares, you're going to have a problem that you need to solve. So it's really good to have a systematic process for doing that. First of all, we need to state the letter denoting the allele, indicating dominant and recessive. Now, there's no rules about what letters you use. Something that makes sense. You want to try and ideally use letters that are unambiguous for the difference between the capital and the lowercase. So do select a letter that makes sense to you and is not ambiguous. So this scenario is going to be about albinoism, and it's a recessive trait. So the capital A is the non-albino genotype, and lowercase a is the albino genotype. So that's the first step. Next thing we need to do is state the parental genotypes. So in this case, the father is heterozygous, so capital A, lowercase a, so they're a carrier. And the mother is albino, so she's a homozygous recessive. Next thing we do is enter the parental genotypes in the Punnett square. So here's our Punnett square. and we're putting our father and our mother. The next thing we're going to do is combine each parental allele with the allele from the other parent. And so then what we can do is we can actually work out what the genotype ratio is. So we've got two choices here for our genotype. So we've got either a heterozygous or a homozygous recessive. They're our two genotypes, and they're 50% each. There's a couple of ways we can express this. We can say 2 to 2, but we can simplify that as to 1:1, and then we say what that ratio is. So the heterozygous to the homozygous recessive. Or we can say it's 50% heterozygous, 50% homozygous. Then we can work out what the phenotype ratio is going to be. And that is going to be, again, the same ratio. So we can say is 1:1, non-albino heterozygous is to albino, or 50-50. And that's how we do it. So what we've done so far is a monohybrid cross. We've looked at a single gene controlling a single phenotypic characteristic, like we just did with albinism. We can also have a look at a dihybrid cross, which is looking at two genes that control two unrelated characteristics. So the two genes that we're looking at, they could be on different chromosomes, or they could be on the same chromosome. Now, if they're on the same chromosome and they're close together, there's a good chance that they will be inherited together. So we can actually have a look at the ratio for the dihybrid cross. So we can actually compare the actual ratio of phenotypes and the theoretical ratio for the dihybrid cross to actually see if it may well be that they're linked together. So let's have a look at what happens when we combine these different alleles in a dihybrid cross to see what the actual genotypic relationships or ratios are. So this is the first gene that we're looking at, and we've got our two alleles, and this is the second gene we're looking at, and the two alleles. So we've got this method called the crab claw method. What we're going to do is use our crab claw to try and identify what all of the different genotypes can be. So first of all, we've got the capital A, capital B, and then the next one we've got is capital A, lowercase b, and then the other way around, we've got lowercase a, capital B, and we've got lowercase a, lowercase b. Okay, so what that does is just identifies for us the different allele combinations for our dihybrid cross. Now we actually need to work through and determine what the genotype and phenotype ratio is. So let's go ahead and do that. We're going to use an example here of pea plants. Then they can either have purple flowers, which is dominant, or white flowers, which is recessive. You see how we've got the same letter and the lowercase for recessive. This is one that I said that was difficult to do, capital P, lowercase p. And we're also having a look at this other characteristic or trait about the length of the stems. Tall stems is the dominant one, and the short stem is recessive. And so we've got a lowercase t and an uppercase T. We could use the crab claw to work out what all of the different genotypes are. So, we've got purple tall, and there's actually 4 different ways that we could get purple tall. So let's kind of explain that. So one of the ones, of course, could be purple and tall. Okay. Another one could be purple short. Another one white tall and white short. Okay. So let's start with white short. That's easy, isn't it? Because it has to be, both of them need to be the recessive. So lowercase p, lowercase t. All right, that's easy. Now, white tall. Okay, so white being recessive needs to be lowercase p. But to be tall, it could be either homozygous or heterozygous. And it's the same thing here with purple short. So purple tall, these are both the dominant. So it could be homozygous for both of them, or it could be homozygous for one and heterozygous for the other. And these are the different ratios. Okay, so all of that. So that's the first thing we need to do. All right, so now that's how we would express all of the different types of alleles. But, if you get a question like this, it's probably going to tell you what the, it will tell you what the genotype is for the parents that you're crossing. So we're going for two heterozygous individuals. So their phenotype is going to be purple tall. And let's work out what's going to happen with this cross. So we're going to use a dihybrid cross Punnett square to do that. So this is probably the hardest part to get this set up. What you need to do for both of the parents, you need to think about what are the possible allele combinations that could be in the gamete. Okay, so remember how we said that this one was heterozygous. So we've got a capital P and we've got a lowercase p. So we have a 50% chance, if you like, that there's going to be a capital P, and a 50% chance it's going to be a lowercase p. So I would do that first. I would put in the capital P in the first two and the lowercase p for the other two. Then what you need to do is you need to split your T, your capital and your lowercase t's. So remember that was heterozygous for tall as well. Okay, so that means that there's an option of a capital T or a lowercase t. So we need the four different options. So we've got a capital T and we're going to match it with a capital P. But the other one we need to do is a capital T and match with a lowercase p. That's why they need to be split. Okay, and so then we're going to do the same thing with our lowercase t's. Right, now in this case, because they're both heterozygous, you're going to do it for this one as well. Okay, then you do your combinations. So from here, we could easily work out a genotype ratio, and we've got our phenotype ratio over here. So we can see that there's 9 purple tall, there is 3 purple short, there's going to be 3 white tall, and there's going to be 1 white short. All right, so this is another type of cross. This time we have got a heterozygous purple tall plant with a homozygous recessive short white plant. And when we do this cross, we have a different ratio. This one is 1, 1, 1, 1. So we've got specific ratios that we see for specific types of crosses. And if we see that the ratio is different to that, it might suggest that there's a linkage between the genes. So that means that the genes that are for two different traits, but if they're located close together on the same chromosome, they have a high chance of being inherited together. And partly that's due to with recombination, where portions of the chromosome are swapped. If they're close together, they're likely to be swapped together. So we say these genes are said to be linked. We can have an idea that we've got linkage if the ratio of phenotypes that we actually calculate are different to the 9331 or the 1111 that we saw. The 1111, which was the cross between the heterozygous and the homozygous.